Welcome to episode 3 of Cell Culture series. Today we looking into media and reagents. Your cells are picky eaters. Feed them wrong, and they'll die. Feed them right, and they'll grow, divide, and survive for weeks, months, even indefinitely. That pink liquid in this flask is not just water and salts. It is a precisely engineered cocktail that mimics the fluid your cells live in inside your bloodstream. Every amino acid, every growth factor, every ion in this bottle exists for a reason. By the end of this episode, you'll be able to walk into a cell culture lab, look at a bottle of media, and understand exactly what's in it, why it's there, and what happens when something goes wrong. Let's decode it, layer by layer. Anatomy of a complete medium. Think of cell culture media like a layer cake. Each layer plays a distinct biological role. You can't skip a layer. Remove one and the whole structure collapses, and your cells along with it. Layer 1. Basal medium, DMEM, RPMI, MEM, glucose as energy, amino acids as protein building blocks, vitamins as enzyme cofactors, inorganic salts for osmolarity and ion balance. Layer 2. Serum, typically FBS, 5 to 20%, growth factors, hormones, lipids, carrier proteins. The secret sauce, and also the most controversial ingredient in modern cell culture. Layer 3. Supplements, glutamine, antibiotics, bicarbonate, glutamine, preferred energy source for rapidly dividing cells. Penicillin, streptomycin, antimicrobial insurance, sodium bicarbonate, pH buffer in equilibrium with carbon dioxide. Let's drill into each layer, because this is where most textbooks stop short, and where real understanding begins. The basal medium. When scientists in the 1950s first needed to grow cells outside the body, they had to reverse engineer what those cells were bathing in inside the organism. Harry Eagle at the NIH spent years figuring this out. The result was Eagle's minimum essential medium, MEM, still in use today. DMEM, Dulbecco's modified Eagle medium, is just a supercharged version with 4 times the amino acid and vitamin concentration. RPMI 1640 was developed at Roswell Park Memorial Institute specifically for suspension cells, lymphocytes, leukemia lines. The basal medium you choose depends entirely on your cell type. Using RPMI for an adherent epithelial cell line, or DMEM for a suspension B cell line, can seriously compromise growth and validity. Glucose in the medium is the cell's primary fuel. Standard DMEM comes in high glucose, 4.5 grams per liter, and low glucose, 1 gram per liter, formulations. Fast-dividing cancer cell lines thrive on high glucose. They are essentially Warburg machines. Primary cells and metabolic studies often require low glucose to avoid confounding your results. Serum, the necessary evil. Here's the honest truth about fetal bovine serum. We use it because it works, not because we fully understand it. FBS is collected from bovine fetuses during slaughter. It contains hundreds of undefined growth factors, hormones, lipids, adhesion factors, albumin for transport, and transferrin for iron delivery, all in wildly variable concentrations depending on the lot. You actually have to heat and activate serum at 56 degrees Celsius for 30 minutes to destroy complement proteins before adding it to media. And every new lot you purchase should be tested against your cells before committing to a bulk buy, because serum composition varies significantly between animals and processing facilities. This undefined variability is exactly why the biotech industry is sprinting towards serum-free, chemically defined media. When you're manufacturing a therapeutic protein or a cell therapy product, you cannot have a biological variable you don't control sitting in every flask. We'll come back to this in the real-world section. pH and osmolarity, the physiological sweet spot. Your cells evolved in a very specific chemical environment. They want to live at pH 7.2 to 7.4. Drop below 7.0 and you're in acidosis. Cell signaling becomes dysregulated, proliferation slows, and apoptosis increases. Go above 7.6 and you're in alkalosis, equally problematic. The pH indicator in most media is phenol red. At the correct pH it is a vivid salmon pink. It turns yellow when acidic, usually from carbon dioxide buildup or bacterial contamination producing acid. It turns purple or magenta when alkaline, usually because the cap has been left loose and carbon dioxide has off-gassed. If your media is yellow before you've even added it to cells, something is already wrong. Osmolarity must stay within 280 to 310 milliosmoles per kilogram. This is isotonic. Go hypotonic and your cells swell and lyse. Go hypertonic and they shrivel, a process called crenation. When you're adding supplements to base media, always account for their contribution to osmolarity. Adding too much glutamine, too much sodium chloride, too many antibiotics, all of it shifts the osmolarity. Glutamine, the unstable workhorse. L-glutamine is the primary carbon and nitrogen source for rapidly dividing cells, especially in the TCA cycle and nucleotide synthesis. But here's the problem. Glutamine is chemically unstable. In aqueous solution at 4 degrees Celsius, it spontaneously degrades into ammonia and pyroglutamic acid. The ammonia is toxic to cells. Shelf life is roughly 4 weeks after supplementation. This is why many labs now use Glutamax, a more stable dipeptide form, L-alanine L-glutamine, that releases free glutamine enzymatically inside cells. If your cells are growing poorly and you can't identify the cause, expired glutamine is always on the short list of suspects. Okay, I'm going to walk you through media preparation exactly as it happens in real practice. Every step matters. Let's go through it. Step 1. Gather and thaw your supplements. FBS comes frozen at -20 degrees Celsius. You cannot add it to base medium while it's frozen, and you should not thaw it at room temperature. Too slow, too much risk of protein aggregation. The correct method is to thaw it in a 37 degrees Celsius water bath, swirling the bottle every 15 minutes to ensure even warming. Total thaw time for a 500 milliliters bottle is typically 45 to 60 minutes. Once thawed, you should heat and activate. Place the bottle in a 56 degrees Celsius water bath for exactly 30 minutes, swirling every 10 minutes. This kills complement proteins that would otherwise damage your cells. After heat and activation, cool to room temperature before adding to media. Penicillin, streptomycin comes as a frozen stock at 100 times concentration, 10,000 units per milliliter penicillin and 10,000 micrograms per milliliter streptomycin. Thaw it at 4 degrees Celsius overnight or briefly at 37 degrees Celsius. Your working concentration is 1 times, meaning you add 5 milliliters to a 500 milliliters bottle of media for a final dilution of 1 to 100. Glutamax also comes as 100 times concentrate and gets the same treatment. Step 2. Work inside the biosafety cabinet. Always run the UV lamp for 10 minutes before I start, then leave only the fan running. Never work under a live UV lamp. Everything that goes into the hood has been surface decontaminated with 70% ethanol. Spray your gloves, spray the bottles, spray the pipette packaging. Aseptic technique is not paranoia. It is the price of admission to cell culture. Step 3. Add supplements to the base medium. Start with 500 milliliters of DMEM high glucose. To this, add 50 milliliters of heat inactivated FBS. This gives a final concentration of 10% V, followed by 5 milliliters of penicillin streptomycin and 5 milliliters of glutamax. Maintain a consistent workflow from left to right. Supplements are placed on the left, the base medium remains in the center, and used pipettes are discarded into a waste beaker on the right. This setup prevents reaching across an open vessel and reduces contamination risk. After each addition, the source bottle must be recapped before opening the next one. Cross-contamination between supplement bottles is uncommon but can happen, and a contaminated stock, especially antibiotics like pen, strep, can affect multiple batches of media over time. Step 4. Filter sterilization. Even though the bottle is already labeled sterile, filtration is still important after adding supplements, especially FBS. Commercial FBS is tested for sterility, but once multiple components are handled and mixed inside a biosafety cabinet, there is always a small risk of contamination during processing. Filter sterilization also helps remove tiny precipitates that can form when proteins and lipids in FBS interact with the base medium at 37 degrees Celsius. The pore size of the filter is critical. A 0.22 micron filter is commonly used because most bacteria are much larger, typically between 0.5 and 5 microns, so they cannot pass through. But one major threat still remains: mycoplasma. Mycoplasma contamination is one of the biggest hidden problems in cell culture. These organisms are extremely small, around 0.1 to 0.8 microns, meaning they can sometimes pass through a 0.22 micron filter. That's why filter sterilization alone is not enough. In any serious laboratory, regular mycoplasma testing is a mandatory quality control step. Step 5. Label and store. Media should never be stored without proper labeling. Every bottle must include essential information for identification and traceability. The label typically contains the media name, such as DMEM complete, the date of preparation, the preparer's initials, the lot numbers of supplements like FBS and penicillin streptomycin, and the expiration date, which is commonly set at 4 weeks from preparation. Prepared media is stored at 4 degrees Celsius and protected from light exposure. Light can degrade sensitive components in the medium, including riboflavin and other photosensitive vitamins. For additional protection, some laboratories store media bottles wrapped in aluminum foil. Quality check. What healthy media looks like. Freshly prepared complete DMEM has a bright salmon pink color, which indicates a healthy pH range of approximately 7.2 to 7.4. The osmolarity is typically around 300 mosm per kilogram, as confirmed by the manufacturer. Now compare that with media stored incorrectly with a loose cap overnight. The color shifts to purple because carbon dioxide escapes from the medium, causing the pH to rise to around 7.8. Cells introduced into this environment can immediately experience alkaline stress, which affects growth and viability. And here is one of the most important warning signs in cell culture: yellow media. This color change usually indicates acid production caused by bacterial contamination. Along with the yellow color, contaminated media often appears turbid or cloudy, unlike clean media which should remain clear. Even a small contamination event, such as contact with a non-sterile pipette tip, can rapidly affect an entire culture. If contaminated flasks are opened inside the biosafety cabinet without recognition, contaminants can become aerosolized and spread throughout the workspace. That is why contaminated cultures must be discarded immediately into designated biohazard waste. Before adding any media to cells, always warm it to 37 degrees Celsius in a water bath for 15 to 20 minutes. Cold media, even at room temperature, causes thermal shock in adherent cells. Their membranes become temporarily less fluid, channels misfunction, and detachment can occur. This is especially critical for sensitive primary cells. Mistake clinic: the media mistakes that destroy cell cultures. Let's talk honestly about the most common media-related mistakes seen in training labs and even in professional research environments. The important thing is this: almost every one of these mistakes is preventable once the science behind it is understood. And in cell culture, small mistakes rarely stay small. Mistake 1. Using expired or degraded media. One of the most common issues is using media that has passed its recommended storage window, especially media containing glutamine. Standard L-glutamine is unstable in solution. Over time, it naturally degrades and produces ammonia as a byproduct. That ammonia becomes toxic to cells and can alter cellular metabolism without any obvious visual warning. This means cells may suddenly grow slower, appear stressed, or produce inconsistent experimental results. In many cases, researchers spend days troubleshooting incubators, serum quality, or cell density, when the real issue is simply degraded glutamine. The solution is straightforward: always check the preparation date before using complete media. Most complete media should ideally be used within 4 weeks when stored properly at 4 degrees Celsius. Many laboratories now use stabilized glutamine supplements such as Glutamax because they remain stable for much longer and reduce ammonia buildup. If unexplained growth problems appear, glutamine quality should always be one of the first variables checked. Mistake 2. Adding cold media directly to cells. This mistake looks harmless, but cells experience it immediately. Media stored in a refrigerator sits at around 4 degrees Celsius. Human and mammalian cells normally live at 37 degrees Celsius. That is a temperature difference of more than 30 degrees. Adding ice-cold media directly onto cells creates sudden thermal shock. Cell membranes become stressed. Metabolism slows abruptly. Sensitive cell lines may partially detach or alter their behavior. Even if the cells survive, experimental consistency can suffer. This is especially important in assays involving signaling pathways, drug responses, or metabolic measurements. The fix is simple but essential. Always pre-warm media to 37 degrees Celsius before use. In experienced laboratories, pre-warming becomes automatic. The moment the biosafety cabinet is turned on, the media bottle is placed into the water bath or warming system. Good cell culture technique is built from habits like this, habits repeated consistently every day. Mistake 3. Storing complete media for too long. Another common problem is preparing large volumes of media that remain unused for weeks or even months. At first glance, the media may still look perfectly normal, but chemically, important components are slowly degrading. Glutamine breaks down, antibiotics gradually lose potency, growth factors and proteins in serum begin to oxidize. Over time, the media becomes less supportive for healthy cell growth. This creates variability between experiments, one of the biggest problems in biological research. Cells may grow well one week, and poorly the next, even when everything appears identical. The solution is smarter preparation planning. Prepare smaller batches based on actual weekly usage. If only 100 milliliters are used per week, then preparing 100 milliliters at a time is often better than making 500 milliliters that will sit unused for long periods. Fresh media produces more reliable biology, and reliable biology produces reliable science. Mistake 4. Over-reliance on antibiotics. This is one of the most dangerous habits beginners develop. Antibiotics are helpful, but they are not a substitute for sterile technique. Many new researchers assume that adding extra antibiotics will make cultures safer. In reality, excessive antibiotic concentrations can damage mammalian cells themselves. High concentrations may interfere with protein synthesis, stress cellular metabolism, and alter experimental outcomes. But the bigger problem is hidden contamination. Low-level contamination, especially mycoplasma, can survive silently when antibiotics suppress visible symptoms. The culture may appear healthy while the cells are already physiologically altered. This leads to inaccurate data, failed reproducibility, and sometimes entire projects built on compromised biology. The correct mindset is this: antibiotics are a safety net, not a crutch. In fact, many experienced cell culturists routinely work without antibiotics. Why? Because clean technique should prevent contamination on its own. If contamination appears immediately in antibiotic-free culture, that is valuable information. It means the workflow needs improvement, not stronger antibiotics. The shared community media bottle. Now for one of the most infamous problems in training laboratories. The shared community media bottle. A bottle left sitting on the bench for days, repeatedly open, sometimes uncapped, used by multiple people. This creates three major problems simultaneously. First, contamination risk. Every opening increases the chance of introducing bacteria, fungi, or airborne particles. Second, carbon dioxide loss. When media sits exposed to air, dissolved carbon dioxide escapes. This shifts the pH upward, turning the media purple and creating alkaline stress for cells. Third, osmolarity changes. Evaporation slowly concentrates salts and nutrients, altering the chemical balance cells depend on. Even if contamination never becomes visible, the media itself may no longer support normal cellular behavior. The solution is disciplined media handling. Label every bottle clearly. Store media in the refrigerator immediately after use. And only warm the amount needed for that specific session. Good laboratories are not built on expensive equipment alone. They are built on consistency, discipline, and respect for small details. Here's why every skill in this episode has direct commercial and regulatory relevance, and why understanding media is not just academic. The FDA's regulations for biologic manufacturing require that cell culture processes used to produce therapeutic proteins, vaccines, monoclonal antibodies, and cell therapies be fully characterized and reproducible. Serum-containing media fundamentally cannot meet that bar. FBS is an animal-derived component with lot-to-lot variability, risk of viral contamination, and supply chain instability. The COVID-19 pandemic exposed exactly how fragile FBS supply chains are. This is why every major biologic manufacturer, Genentech, AstraZeneca, Regeneron, and the cell therapy space, has invested massively in proprietary serum-free, chemically defined media. When you use a biologic drug today, an mAb, a CAR T-cell therapy, a recombinant vaccine, there is a very high probability that the cells that produced it or that drug were grown in a defined, serum-free medium. Every component is listed, characterized, and present in precise concentrations. Companies like Thermo Fisher Scientific and Millipore Sigma, Sigma Aldrich's parent, generate billions of dollars annually from life science media and reagents. Thermo Fisher's cell biology division alone reports multi-billion-dollar revenues from media, sera, and supplements. This is not a cottage industry. This is the backbone of the entire modern biotech supply chain. So if you're thinking about career paths in biotech, formulation scientists who design new media, process development scientists who optimize media for scale-up bioreactors, and QC scientists who validate each media lot are all in continuous demand. Your understanding of what's in that bottle is a genuinely marketable skill. Career note: process development roles at biotech manufacturers, optimizing media and feeding strategies for bioreactors, routinely pay $80,000 to $130,000 plus USD at entry to mid-level. Cell culture media formulation is a specialized, quantitative skill. If you can explain what's in that bottle and why, you can walk into an interview and immediately differentiate yourself. Let's recap. Media is artificial blood. Every component mimics the physiological environment your cells evolved in. Nothing is in there by accident. Serum provides growth factors. Its replacement with defined, serum-free media is the central challenge of modern biomanufacturing. pH 7.2 to 7.4 and osmolarity 280 to 310 mosm per kilogram. These two parameters are non-negotiable. Phenol red is your real-time readout. Trust it. Next episode, we're getting into passaging, how to subculture your cells without destroying them. If you enjoyed this video, found the topic helpful, or believe this content could be useful to others, please take a moment to like, share, and leave a comment below. And don't forget to subscribe to the channel for more videos like this. Thank you for watching.